Okay, so let's start learning about microbiology using the most basic questions. To make things a little more interesting, let's start with when and who. Okay, when did microbiology come to be, or when did microbiology start, and who are to blame for adding such a challenging subject to medicine? For thousands of years, ancient medicine believed that it was all about liquids called humors. I'm sure you've heard of the terms vitreous humor or aqueous humor. Okay, so these are liquids in our body. And that some diseases were caused by an invisible badness or evil in the air, or miasma. So the miasma theory believed that diseases were caused by foul-smelling airs that came from rotting meats or swamps and other putrefying sources. Of course, they did not yet know at that time that the smells were only a byproduct of microbes eating organic matter. And for a long time, they believed in the theory of spontaneous generation when they observed maggots to come out of nowhere in rotten meat. This was also actually promulgated by Aristotle. Of course, this would later be debunked by a number of experiments done by later quote unquote rebel scientists. During around the 1660s, an Englishman by the name of Robert Hooke, I'm sure the name sounds familiar to you from your elementary or high school biology. So, Robert Hooke described for the first time cellular structures in tree bark and drew sketches of little structures that seemed to be alive. He then became known to have discovered the cell, specifically plant cells. Later, in the late 1600s, a man, a tailor, merchant, and a lens grinder from the Netherlands or Holland, meaning he was Dutch, by the name of Anthony van Leeuwenhoek, out of curiosity, made glass lenses of his own and began asking, what does it really look like of everything around him? Whether it be the stinger of a bee, the brain of a fly, the leg of a louse, a drop of his own blood, flakes of his own skin, etc. No? Until one day, he turned a lens onto a drop of water from the pond. Well, we don't really know what he saw, but according to his letter to Royal MD, joke, to his letter to the Royal Society of London, he wrote some green streaks, spirally wound serpentwise and orderly arranged. Among these, there were, besides, very many little animalcules. Some were round, while others a bit bigger, consisted of an oval. On these last, I saw two little legs near the head and two little fins at the hindmost end of the body. And the motion of most of these animalcules in the water was so swift and so various, upwards, downwards, and roundabout. That was wonderful to see. Later, interestingly, he came about looking into the plaque of someone else's teeth after he looked into the plaque of his own teeth. So, in another letter, he wrote, the number of these animals in the plaque of a man's teeth are so many that I believe they exceed the number of men in a kingdom, in a quantity of matter no bigger than 100th part of a grain of sand. Nagmamahal, Anthony. Well, it is because of this that he is known as the father of microbiology and protozoology. So this goes to show that curiosity kills the cat. No. Curiosity is the father of invention. Mali pa rin. So jokes aside, curiosity makes you find a lot of interesting things to do. It keeps leading us down new paths. Well, that's according to Walt Disney. And according to Richard Duke, it is the forerunner of discovery. But yet, both of them still did not know that they were connected to disease of humans. It was only in the mid 1800s when scientists, quote unquote rebels, coined the germ theory of disease. So they believed that pathogenic microorganisms or germs caused infectious disease. It was during this time when the cholera pandemic spread throughout the world. In 1854, there was a cholera outbreak in Soho, London, killing around 600 people. There, one English physician had a hunch of how the disease might have broke out, and he mapped the disease occurrences in the area and found out that many of the occurrences clustered around an area on Broad Street that had one thing in common. The source of the water of the people was a pump, or the pump in Broad Street. So he hypothesized that the organism causing cholera might be multiplying in water. This doctor became famous for removing the pump handle so that the people would not be able to get their water from that pump. And so they were forced to get their water from somewhere else. Amazingly, the cholera cases in that area decreased. So that physician was actually Dr. John Snow. He is now known as the father of epidemiology for what he did. So he is the same Dr. John Snow who might have been introduced to you already in your PCM research subject. Something nice to know about him is that he was also known for popularizing the use of chloroform as anesthesia during childbirth, and in fact used it with Queen Victoria when she delivered at least 3 of her children. But this really didn't prove germ theory, and scientists would require a better model of what germs are and how they behave. So one of the first scientists to test the theory of spontaneous generation, or abiogenesis, was Francesco Redi, using a simple experiment. So he placed meat in 2 different jars and covered one of the jars with fine gauze. So this prevented flies from landing on it, and he left the other jar uncovered. Flies surrounded both jars, but he observed that maggots only appeared on the meat in the uncovered jar. The flies laid eggs on the gauze covering the second jar, and maggots appeared on the gauze but not on the meat. So this proved that the meat was not the source of the maggots. So this and other similar experiments put to rest any ideas that maggots, mice, and other complex organisms arose spontaneously. But the scientific community was not easily convinced. No? Even Theodore Schwann, the father of histology, who defined the cell as the basic unit of animal structure, he actually maintained that forces in the air were the source of life. Later on, in 1857, a French chemist observed that microscopic yeast on grape skins makes grape juice ferment into wine. And when he sterilized the skins, he noticed that fermentation did not happen. So when we say fermentation, we are referring to the conversion of sugars into alcohol or ethanol and carbon dioxide. So this chemist made an analogy that the microbes that made wine and beer were similar to the microbes that causes infections in humans. So he then hypothesized that microbes were present everywhere and were responsible for all kinds of phenomena, like, for example, causing food to spoil. That person was Louis Pasteur, who is now considered the co-founder of microbiology and the founder of zymology, or the science of fermentation. So later on, in 1865, he patented a process in which liquids, such as milk, were heated to a temperature between 60 to 100 degrees Celsius to kill the microbes present there. And this became known as boiling. Joke. Became known as pasteurization. So he also later partnered with a British surgeon, Dr. Joseph Lister, to develop hygienic medical practices. One of them, the principle of sterility in the operating room, which started with sterilization of instruments. He also did an experiment to disprove the theory of spontaneous generation using modified glass flasks, and proved that living microbes that grew in beef broth were not spontaneously generated. And from his observations on grapes, he reasoned that living things can only grow from other living things, or biogenesis, or life from life. Mr. Pasteur also then worked on immunology and vaccination and concluded that if you could identify a microorganism, you could give a weakened version of it to someone, and he would develop immunity to the real microbe. So, this is the principle of active vaccination that we're using right now. Between the 1880s and the 1890s, a German physician named Robert Koch established bacteriology in a laboratory in Berlin and focused on studying microbes that were particularly related to human health and disease. He actually traveled to places with epidemics such as Egypt and India to try to identify the microbes causing the disease there in the hope to create vaccines. So he is also known to have identified the germ that causes anthrax and later on the microbe that causes tuberculosis. His team was also credited to have identified a lot more microbes. For example, those that caused diphtheria, typhoid, pneumonia, gonorrhea, meningitis, tetanus, leprosy, syphilis. So imagine, no, if he passed on his autographed microscope to you, unsterilized. So these events only marked the beginning of our understanding of microbes and the diseases that they can cause. And we have not stopped learning and discovering. We even have learned the ability to study genomes and even harness biological processes. But don't worry, we will just be focusing on the basic and important for now, those that will be relevant to your practice of medicine a few years from now. Okay?